Assalamu alaikum everyone. Today inshallah we're gonna look at how multiple polypeptides could be synthesized in bacteria and eukaryotes at the same time. So once the mRNA is available for translation, multiple ribosomes at the same time can bind to the mRNA to use it for building a polypeptide. Okay? So we have a cellular structure observed under the electron microscope called polyribosome or polysome. So polyribosome or polysome consists of multiple ribosomes binding to the same mRNA and building polypeptides using the codes of these mRNA at the same time. So, and as you can see in this cartoon, we have 4 ribosomes bound to the same mRNA. Of course, some ribosomes are already in advanced stage of translation. So this one ribosome is about to finish already synthesize the completed polypeptide. This one just started. These ones are still adding, reading the codons in the mRNA and adding one amino acid at a time. Okay. And here at the bottom we have a real electron microscopy, transmission electron microscope image showing one mRNA. Okay, we could draw a line like this, and multiple ribosomes. So this is the mRNA, for example, and these are the ribosomes that are bound to this mRNA, actively synthesizing, for example, polypeptides. So all of these ribosomes will synthesize the same polypeptide because they are using the same codons on this mRNA. Okay. Now, remember our discussion at the beginning of this chapter about the differences between gene expression in prokaryotes and eukaryotes. In eukaryotes, we have the nucleus. And inside the nucleus, we have the DNA is transcribed into pre-mRNA through transcription, and then the pre-mRNA is converted into a mature RNA by RNA processing. Then the mRNA is basically in the cytoplasm, available for the ribosome, and through translation we have a protein, a polypeptide is generated. So this is just a quick summary of the events that happens during the gene expression in eukaryotic cells. Okay, so this is the nucleus. Here we have the cytoplasm. Of course, here we have translation. Sorry. In prokaryotes, so now we have, let's say, physical separation between the translation and the transcription. Transcription happens in the nucleus, translation in the cytoplasm. In prokaryotes, for example, in bacteria, an example prokaryotic cell. Translation and transcription both happens at the same time. So we have something called coupling of translation and transcription. تزاوج or مش تزاوج جمع between transcription and translation. So here we have a DNA, a gene that is being actively transcribed by multiple RNA polymerase. And I showed you before that how different or multiple RNA polymerases can bind to the same gene, actively transcribing that gene at the same time. So as the mRNA is being synthesized in bacteria, because there's no nucleus and the DNA is not inside the nucleus separated from the cytoplasm, ribosomes will immediately bind to the emerging mRNA and start actively synthesizing the polypeptide. So as you can see here, we have a polysome, polyribosomes or polyribosomes, and we have multiple ribosomes actively transcribing the same gene, and multiple ribosomes actively translating the same RNA transcripts. Remember, in prokaryotes, we have only transcription and translation. There is no processing. Transcription and translation. No RNA processing. Okay? That's why transcription and translation in eukaryotes could be coupled, and this could not be done inside eukaryotic cells. Okay. So this figure summarizes what we've just learned so thus far in this chapter. So the last topic in this chapter is about mutations. And here we're going to learn about small-scale mutations. So let me give you just an introduction about the different types of mutations in general. So mutations in general could be classified based on their effect or the size of their effect into small scale mutations and we have large scale mutations. So large scale mutations means we have, and we're gonna discuss large scale mutations in another chapter, but for now, let me just introduce this term. We have amplification of a certain region within the chromosome, making more copies of a certain region within the chromosome. Here we're talking about mutations at the chromosome level. So these are the types of mutations that affect large regions within the chromosome. We have also deletion. Deletion means amplification. In Arabic means amplification مضاعفة. deletions means إزالة. So large regions within certain chromosomes are deleted or removed. We have translocation. Again, we're going to talk about these large-scale mutations in another chapter. This is when one region of a chromosome is exchanged with another region of another chromosome. Inversion, this is when one region of the chromosome is changes its direction. Okay? But small scale mutations, these are changes. We have, for example, point mutation. Okay, and these point mutations are simply changes in a single nucleotide. We have also small deletions. You're talking about deletion of a couple of amino acids compared to large-scale deletion, deletion of let's say thousands of nucleotides. Here we're talking about few nucleotides, 1, 2, 3, or so, or small insertions. Here we have also insertions. Deletions and insertions, they come together typically. Insertions means إضافة. Okay. Inversion means عكس. So small insertions, insertion of a couple of nucleotides, maybe 1, 2, 3, 4, 5, 6, few, just a few. Now, point mutations, in terms of their effect on the amino acid sequence, if they happen in a coding sequence or in a codons, in the codons, we have several types of point mutations based on their effect on the amino acid sequence. We have silent mutation, we have missense, and we have nonsense. Nonsense. Okay, so we're gonna today, inshallah, we're gonna learn about small scale mutations. Large scale mutations, we're gonna look at these in another chapter. Okay. So mutations are changes in the genetic information, changes in the sequence. This change could be small change like small scale mutations, or could be dramatic, very big change, just like these that happens at the level of the chromosome. Now, any change, if we're talking about small scale mutations like point mutations, these are just changes in one nucleotide pair of a gene. A change of a single nucleotide could have a dramatic effect or a significant effect at the level of the amino acid sequence and at the level of protein structure and function, and it might lead to the production of an abnormal protein, and this abnormal protein, غير عادي, it might lead to a genetic disease or a disease. So take, for example, individuals that have sickle cell disease. So the beta, hemoglobin, the protein hemoglobin, if you remember, so what's the function of hemoglobin? What is the function of hemoglobin? Hello? Yes, it's responsible for transferring oxygen in our blood. Hemoglobin consists of two types of polypeptides, two beta polypeptides and two alpha. Now, the beta globin gene, and these two polypeptides, the beta is encoded by a beta gene, and the alpha is also encoded by a different gene. The beta polypeptide consists of 147 amino acids. Okay? Now, in people with wild-type beta globin gene, imagine this is just part of the gene. Again, the gene is very large, but here we're showing you codon number 6. Okay, this is codon number 6, and this codon, so this is codon number 6. In DNA, this is codon, the same codon on the mRNA, and codon number 6 encodes the amino acid glutamic acid. So basically, if we're talking about a gene that will result in the formation of, let's say, a polypeptide with 147 amino acids, we will have at least 147 multiplied by 3 codons. This is just codon number 6. Okay, this is in normal people. Okay, so all normal people, we have this codon, they have this codon number 6, and normal beta globin and normal hemoglobin will have glutamic acid as the number 6 residue in the beta globin polypeptide. Now, in individuals with sickle cell disease, their beta globin has a point mutation. Point mutation that result in a substitution of one base pair. So instead of having TA, they have AT, which changes the codon number 6 from GAG into GUG, and the GUG encodes the amino acid valine. This single amino acid, this single nucleotide substitution leads to a single amino acid substitution. So here, so going from here to here, we have single base pair substitution. Substitution means tabdil. Single one, base pair. This is a base pair, this is a base. And here, this single amino acid substitution causes an amino acid substitution. So instead of having, this is codon number 6. Okay, so instead of having glutamic acid as the number 6 amino acid as glutamic acid, people with sickle or individuals with sickle cell disease, they will have valine as residue number 6 in their beta globin polypeptide. And this single amino acid substitution caused by this single base pair substitution is enough to cause sickle cell disease. So as you can see, substitution of only 1 out of 147 amino acids is enough to cause a disease, or the substitution of 1 base pair out of maybe, I don't know, 500 base pairs is enough to cause a disease. Okay? So as you can see, sometimes in some cases, a single amino acid base pair substitution might have a dramatic effect on the function of the protein encoded by some genes. Now, point mutations, there could be single nucleotide pair substitution like this, or there could be nucleotide pair insertions or deletions. Let's talk about the first one. So a nucleotide pair substitution replaces one nucleotide and its partner with another pair, like here. This is replacing this pair with this pair. Now, in terms of the effect on the amino acid sequence, the effect could be having a silent mutation, which means the amino acid will not be changed, or missense mutation, or nonsense mutation. So take, for example, here we have a DNA template consisting of 1, 2, 3, 4, 5 codons. This is the mRNA consisting of 5 codons. And this mRNA, if it's translated, it will generate this polypeptide, methionine, lysine, phenylalanine, glycine, and here we have the stop codon. This is the normal wild-type DNA sequence. Now, imagine if we have a substitution that happens to this base pair at the level of the DNA. When we talk about mutations, we talk about changes that happens at the level of the DNA, which will be reflected as sometimes, in some cases, changes at the level of the mRNA and then the amino acid sequence. Now, instead of having G and C, now we will have A and T. But now notice that there will be a change in the mRNA sequence. So instead of having C here, we have U. But in terms of the polypeptide, it is the same. Why? Because of redundancy, tikrar, in the genetic code. Remember that some amino acids, or the majority of amino acids, they are encoded by more than one codon. So here we have this codon, GGC, GG. So if we looked at the codon table, let me go back and open the codon table. So GGC and GGU, along with, where is the codon table? Yes, here it is. So GGU, GGC, GGG, and GGA, all of these, they encode the amino acid glycine or glycan. That's why this is called the silent mutation, silent. It did not have any effect. There is a change at the level of the DNA, a change at the level of the mRNA, but there is no change at the level of the polypeptide sequence. Okay. All right. So let's talk about the other types of the other effects of nucleotide pair substitution. Here we have the same normal DNA. We have the codons in the mRNA and the nucleotide, the polypeptide sequence. Imagine if we have here a nucleotide pair substitution, and instead of having CG in the DNA and G in the mRNA, we have TA and A at the mRNA level. Now, what will happen is that this nucleotide pair substitution will change the first nucleotide in this codon, encoding glycine, into this codon. So now GGC encodes glycine, but now GGC is changed into AGC, and AGC encodes serine. So now we have a change in the nucleotide, in the polypeptide sequence. Instead of having methionine, lysine, phenylalanine, glycine, now we have methionine, lysine, phenylalanine, serine. This effect is called, this mutation is now called missense mutation. Okay? We have another type of nucleotide pair substitution called nonsense. Again, this is the same normal or wild type DNA sequence, wild type mRNA sequence, wild type amino acid sequence. Imagine if there is a nucleotide base pair substitution here at this loci, at nucleotide number 4. So instead of having TA, now we have AT. And instead of having A on the mRNA as the 4th nucleotide, we have U. And this U will change the first nucleotide in the second codon from A to U. And now the second codon is not AAG, it is UAG. And UAG does not encode any amino acid. It's a stop codon. So now, instead of having a polypeptide consisting of 4 amino acids, we have a polypeptide with just only 1 amino acid, which is the methyl. This type of mutation is called nonsense. Okay? So, point mutations could be silent, missense, or nonsense. But remember, these happens if we're talking about nucleotide pair substitution. So, point mutations, we have 2 kinds. We have single nucleotide pair substitutions, which could be silent, nonsense, or missense. خلينا ناخذ بريك 5 دقائق ونرجع على 2:05. So we've discussed silent, nonsense, missense, single nucleotide base pair substitution. Now we're going to discuss nucleotide pair insertions or deletions, insertions إضافة, and حذف deletions, and how they might have an effect on the amino acid sequence. So here we have the same nucleotide sequence with the same codons we have been discussing so far, the codons and the amino acid sequence. Imagine if here, let me use a different, here, a nucleotide is added. a nucleotide pair is added. Okay, here. This nucleotide pair is AT. So between this C and between this T. So this is C number 3 nucleotide in the order of this DNA template, and this is nucleotide number 4. Here is nucleotide number 3. Here is nucleotide number 4. Notice what will happen is that we have a change in the codon sequence at the level of the DNA and at the level of the RNA. So the first codon was AUG, encoding methionine. Now it's AUG, still AUG. The second one used to be AAG, and now it is UAA. And UAA, it does not encode any amino acid. It's a stop codon. So now what we have, what we call a frame shift. A frame shift means we have a change in the codons that are read by the ribosome. So here, for example, so this is nucleotide base pair insertion. In nucleotide pair deletion, imagine if this nucleotide pair is deleted, is removed. Okay, so here we have nucleotide, let's say number 8. This is 9. This is 10, and now we have only A is missing. So this is number 8 and 10, and 9 is missing. This is again will have an effect on the codons. So the third codon used to be UUU, but because this U is removed, now the third codon will be UUG, because this one is gone. Now we have UUG, and the second codon will be affected because the first codon here of glycine is now added as the third nucleotide of the codon of this codon of the third codon. So the fourth codon, the first nucleotide of the fourth codon is added as to the third codon, or is added as the third nucleotide of the third codon. So now we have what we call a frame shift causing missense. So this could continue over and over and over. So all the nucleotide sequence beyond, or the amino acid sequence beyond the second code, the second amino acid will be changed. Okay, so this is what we call a frame shift. So what's a frame shift? So we have something called open reading frame, or ORF for short. This is the open-reading frame. This is the normal open-reading frame. You have the first codon AUG, the second codon AAG, UUU, GGC, UA. This is the open-reading frame, and this is the open-reading frame for at the level of DNA. A frame shift is caused by deletions or insertions. So the shift, there is a shift in the open reading frame. Instead of reading this one as the first codon, this one as the second codon, and this one is the third codon, no. Now the third codon consists of this A, and this C. So now we have a shift in the codons. Okay? This is what is meant by shift. Shift يعني in Arabic, what does shift mean? إزاحة, maybe we could say إزاحة. Okay? So as you can see, both nucleotide pair deletions or insertions can cause, in some cases, frame shifts. Now here, the frame shift introduces a new, basically an early stop codon. Here, the frame shift will change the amino acid sequence of the polypeptide beyond the second amino acid, for example. This could continue until we have a stop codon. Okay, or a stop codon here. So these dots indicates that reading of this mRNA will continue over and over until there is a stop codon. Usually the stop codon is the 5th codon, but maybe here, since there is a frame shift, now there is no longer a stop codon, and reading of the mRNA transcript will continue until another stop codon will be encountered by the ribosome. So this is the last part of this chapter. So in the previous chapter, we learned about the steps of gene expression. In this chapter, we're going to learn how gene expression is controlled or regulated. So here we have تنظيم التعبير. And we're going to start looking at the main mechanisms of gene expression in eukaryotes and prokaryotes. So organisms, they have in their genomes genes, and these genes encode products. But not all genes are expressed all the time. Some genes are expressed all the time. Some genes are only expressed in specific situations or in a specific type of cells or at specific time within the life of the organism. So organisms, they regulate gene expression sometimes in response to environmental conditions. Again, the expression of genes depends on many factors. Some genes are expressed all the time. Some genes in all types of cells. Some genes are only expressed in certain types of cells. Some genes are only expressed at certain times of the life of the organisms. And some genes are only expressed in certain environmental conditions. ظروف بيئية. Why do we care about studying gene expression? Because understanding gene expression allows us to understand how organisms develop from, for example, how eukaryotic cells or multicellular eukaryotic organisms, how they develop from a single cell into a whole organism. Because gene expression is responsible for the development of an organism, a eukaryotic organism, from a single cell into a whole organism. Also, gene expression, as we will learn later on this chapter, allows the generation of different cell types, different types of cells. Okay. In prokaryotes, gene expression is important to allow the bacteria to respond to its ever-changing environment. So bacteria living in the environment will continuously change its gene expression according to the condition outside in the environment. Or as the environment changes bacteria outside the bacteria will change its gene expression accordingly. Okay. So we're gonna first learn about some of the major mechanisms by which bacteria can change its the expression of certain genes. And when we talk about change in gene expression, we will be discussing how genes could be turned on, then it could be turned off, or maybe a gene that is turned off its expression is turned off, or a gene that is not expressed will be expressed, or maybe change in the level of expression, in the rate of expression. Okay. So here we're talking about switching on off gene expression, or maybe changing the level of gene expression. So we have switching. So changing changes, let's say, in gene expression. We're gonna discuss two major switching on, off, the expression of genes, or we're going to talk about changing the rate of expression. Increase the rate, increase the level of expression, or decrease the level of expression. Okay? In prokaryotes, like bacteria, they have always to control the expression of genes because they are living outside in the environment, and the environment is always changing. So they have to respond to changes in the environment to be able to survive. These responses are actually eventually lead to changes in gene expression. So first, we're going to learn about the control of gene expression in prokaryotes by looking at some examples. So we have in our intestine, in the human intestine, we have, let's say, bacteria living in our intestine, and this bacteria is called E. coli. Okay, so these are bacteria called Escherichia coli, for short, just we say E. coli. So if we look at how E. coli, so if we zoom in here, we have this E. coli cell. So this is an E. coli cell. So E. coli requires, for its survival, the amino acid tryptophan. So we have tryptophan. Tryptophan is an amino acid is important for the survival of E. coli. Now, E. coli inside our intestine, so this is the human intestine, let's say. So this bacteria lives inside our intestine. So for example, if you eat a meal rich in amino acids or proteins, tryptophan will be available for the bacteria. So tryptophan will be picked up by this bacteria, and now the bacteria basically will take this tryptophan and use it to make proteins. So the bacteria will take tryptophan from the environment in the intestine. So here we have the bacteria is surrounded by the intestinal, let's say, environment. environment بيئة. Okay, so the environment surrounding the E. coli is rich in tryptophan, so tryptophan will be picked up by the E. coli. So the E. coli will obtain tryptophan from the intestinal environment. But imagine if you are fasting, you're not eating, okay, and there is no tryptophan. Okay, so no tryptophan. No tryptophan available for the bacteria in the intestine, your intestine. What will happen? The bacteria needs this amino acid tryptophan, otherwise it will die. So the bacteria has to rely on itself. The bacteria in its chromosome have some genes. So these are, let's say, genes important for tryptophan synthesis. So these genes will be used by the bacteria to synthesize tryptophan. So these genes will be expressed if there is no tryptophan in the intestinal environment in order to be able to synthesize tryptophan. But if there is tryptophan in the outside environment, these genes will not be expressed. So if there is a high concentration of tryptophan in the intestine, inside the intestine, okay, tryptophan genes, let's call them, will not be expressed. Why? There is no need to express the genes if there is a high concentration of tryptophan, because the bacteria can obtain tryptophan from the intestinal environment. But if the tryptophan concentration in the intestine is low, or there is no tryptophan, tryp genes will be expressed to make tryptophan. Okay? So this is a classical example where the bacteria will change its expression of these tryptophan genes according to the outside environment, according to the availability of tryptophan. If it's available, there is no need to express the transcription of these genes, or the expression of these genes is turned off. Okay? So expression here is considered off. If there is no tryptophan, expression here of these genes will be on. Okay? So we have a switch on-off of tryptophan synthesis genes according to the outside environment, whether this environment has tryptophan or not. Now, tryptophan can quickly switch on or off the expression from these tryptophan genes by two ways. So tryptophan synthesis from a precursor requires the products of 5 genes: tryp A, tryp B, tryp C, tryp D, tryp E. Trip B and trip A, they will both basically produce polypeptide B and polypeptide A, and both they will form enzyme 3. E and D will both make enzyme 1, and enzyme 2 is only encoded by 1 gene. So here we have 5 genes encoding 3 enzymes, and 3 enzymes are required for this biosynthesis of tryptophan. Now, if tryptophan concentration inside the cell is high, this means if the concentration is high inside the cell, maybe because there is tryptophan outside the cell, okay? We have 2 mechanisms that will basically shut off or switch off any synthesis of more tryptophan. So if we have high concentration of tryptophan inside the cell or inside the bacteria, the high concentration of tryptophan means that some tryptophan will go and bind to enzyme A, and once that happens, enzyme A will be inactive. Now it is inactive, which means that this will not be produced, this will not be produced, and no more tryptophan will be produced. This process is called feedback inhibition. التغذية الراجعة المثبطة. Okay? So in feedback inhibition, within a metabolic pathway, this is a metabolic pathway consisting of 1, 2, 3 biochemical reactions catalyzed by 1, 2, 3 enzymes. The final product concentration will determine whether this metabolic pathway will continue or not. In this case, the high concentration of the final product here, which is tryptophan, will inhibit the first enzyme in this metabolic pathway, catalyzing the first reaction. In this case, it's enzyme 1. Also, tryptophan will inhibit the expression of these genes because there is no need for more expression of these genes, and there is no need to make more of these enzymes because the concentration of the tryptophan inside the cell is high. So here we have 2 types of responses that will basically determine if the cell needs to make more tryptophan or not. If the concentration of tryptophan is high, this means inside the cell, this means no need to make more tryptophan like this. So this could be done by inhibiting the enzymes that are used to make tryptophan. Also, this means no need to express the tryp genes like this. So tryptophan itself can inhibit the enzymatic activity used to make more of it. Tryptophan itself can also inhibit the expression of the genes, the tryptophan genes. Okay. The regulation of enzyme activity by feedback inhibition, this is a short-term response. It's a very quick response. So the enzymatic activity of enzyme 1, 2, and 3, okay, it's controlled mainly by the concentration of tryptophan inside the cell. If it's high, the enzymatic activity of these, or the enzyme activity of enzyme 1 will be decreased, so no more tryptophan will be. But also we have a longer term regulation of enzyme production. So no more enzymes will be produced if the concentration of tryptophan is high. Okay? So if the tryptophan concentration inside the cell is low, this means that this metabolic pathway will be active, these enzymes will be active, and more expression of the tryptophan genes will be happening. Okay? So this is just an example of how bacterial cells, they can regulate the expression of genes according to the conditions, environmental conditions, or outside conditions they are experiencing. Is it clear so far? Do you have questions about the regulation of tryptophan synthesis? Questions? So, as you can see, these 5 genes, so we're talking about 5 genes, 5 different genes. They are all important for tryptophan biosynthesis, which means, in this case, the regulation of these genes must be happening at the same time. So these 5 genes must be switched on, off, if the concentration of the tryptophan inside the cell is high, and their expression must be turned off, just like a light switch you have on and off, if the concentration of tryptophan is low. So we're talking about switching off the 5 genes at the same time if tryptophan concentration in the environment of the intestine is high, or maybe the tryptophan concentration inside the cell is high. So it will be inside I, and here tryptophan inside the cell is high, is low. When the tryptophan concentration outside the cell is low, it will be low inside the cell, and tryptophan genes, the 5 genes, all of them, will be, the expression of these 5 genes will be turned on. Okay? So the bacteria, in bacteria, these 5 genes will be switched on and off at the same time, because these genes will be part of what we call an operon. So these 5 genes will be grouped together, will be located within the bacterial genome like this, very close together. Okay? And this cluster of related genes that are functioning, that have the same function or functioning in the same pathway, is called an operon. So an operon is basically a cluster tajammu of genes in order to control their expression on and off at the same time. So here, for example, these are the 5 genes. This is the operon. These are the 5 genes important for making the 3 enzymes required for the biosynthesis of tryptophan. Now notice that these genes are located back to back within the same region. This is called the TREP operon, the tryptophan operon. It consists of the 5 genes. It consists of a promoter. Now the promoter region contains the RNA polymerase binding site and contains what we call a trip operator. This is like a light switch. This is the switch that will turn on or off the gene. So an operon consists of genes. An operon consists of the genes, a promoter, and an operator. So these are the genes that will give us the products. Okay, for example, the proteins or the enzymes. The promoter, this is where the RNA polymerase will binding site. And the operator is the binding site for a protein called repressor, المثبط. The function of the repressor is to switch off the expression of the genes. Okay, so now each operon consists of a promoter, gene, the genes that are within this operon, and the operator. Here, the genes that we are talking about, in our example, we have the trip A, trip B, all the way to trip E. The operator, we have the trip operator, and the repressor, we have the trip repressor. And the promoter, of course, this is where the RNA polymerase will be. Okay? So here we have the trip operon, the 5 genes, the operator, and the promoter. And notice that the operator is between the promoter, the RNA polymerase binding site, and the genes. Okay? And then we have another gene that is far away from the operator, that is not part of the operator. It's called the regulatory genes, one of them. The regulatory gene is not part of the operon. It has its own promoter. If this gene is expressed, it will produce an mRNA, and the mRNA will produce a protein called the repressor. So this is the trip repressor encoded by the TRPR gene, R for repressor. Okay? Now, if tryptophan is absent, the repressor itself, so now what I'm going to explain is this black arrow. How does the tryptophan affects the expression of these 5 genes? Okay? So, what I am about to explain is how this tryptophan, a high concentration tryptophan, will regulate or will switch off the expression. The minus sign here means negative, or negatively affect the expression of these genes, which means switching off the expression of genes, these genes. Now, the TRPR produces the protein called tryptophan trip repressor. So this is the trip repressor. The trip repressor is inactive, but if there is a tryptophan, if the tryptophan is present, which means tryptophan concentration inside the cell is large or high, the tryptophan itself can bind to the TRP repressor, and now this is inactive by itself. But with tryptophan like this, it will become active TRP repressor, and the active TRP repressor binds to the TRP operator like this. Now the TRP operator, as you can see, binds between the binding site of the RNA polymerase. So this is the RNA polymerase, and between the genes. So now the RNA polymerase cannot move because now the TRP operator is occupied by the TRP repressor, the active TRP repressor, and the tryptophan, and this will block the RNA polymerase from initiating transcription. So there is no mRNA made. So the operon is off. No transcription of the operon genes. No transcription of the TRP genes, which means there will be no production of the enzymes encoded by these genes. In this case, tryptophan is called a co-repressor. Co means متعاون. It will help the repressor, because the repressor itself, without tryptophan, it will be inactive. So if there is no tryptophan, so tryptophan concentration inside the cell is very low, there will be no tryptophan to bind to the TRP repressor. So the TRP repressor will remain inactive. It cannot bind to the TRP operator. There is nothing bound to the TRP operator. There is nothing blocking the RNA polymerase. So the RNA polymerase will transcribe the genes to produce one single mRNA. This single mRNA will have a start codon and a stop codon for each of these genes. So one mRNA, following translation, it will be used to make 5 polypeptides. So this is one mRNA, one mRNA molecule containing the codons for 5 polypeptides. 5 polypeptides. And these 5 polypeptides, these two will form one enzyme, this one enzyme, and these two will form one enzyme, like this. So this is an example where, this is the first example we see one single mRNA that can be translated into 5 polypeptides. So for each, for the codons of each gene, there will be they're going to be flanked by a start and a stop codon. So this is start stop codon for the first gene, followed by the green is start codon and the red stop codon for the second gene, start stop codon for the third gene, start stop codon for the fourth gene, start and stop codon here for the last gene. So here we have a stop codon. Okay. So the operon expression will be on if there is no tryptophan. The operon expression will be off if there is tryptophan. Okay. So this is regulation of expression of the TRP operon. Of expression from the TRP operon